as Brian kind of mentioned, Bio-Techne is a large company. We have years of experience in both developing, manufacturing, and supplying high-quality reagents, anything from proteins to antibodies and so on and so forth. We have over 3,000 employees, more than 750,000 quality products. Thank you. This looks good now?
Yep.
And we also do precision diagnostics. And we also have over 500,000 user citations. In addition, we do have 7 analytical platforms, one of which, the Paula, is going to be the focus for today. But if you want information about any of the other instruments, feel free to ask that as well. So I will be talking about the importance of isolating single cells and how Paula has overcome a lot of the challenges that people have had that are associated with traditional methods of single cell isolation, which are FACS and limiting dilution pipetting. I'm also going to share some performance data that we have generated in-house, as well as a little snippets from our actual customers from their papers, how they're using it specifically in cell line development. And then towards the end of my session, I will also give some very high-level tips on how you can also ensure the optimal outgrowth of your single-cell clones. So single-cell isolation is a technique that is used in various workflows and applications, some of which are listed here. For example, biologics manufacturing, cell and gene therapies, drug discovery, and disease research all involve generating stable cell lines. Single-cell omics like DNA and RNA sequencing and proteomics are becoming more and more of importance, especially in fields like cancer research. And antibody discovery and prenatal diagnostics rely on being able to find and identify and isolate very rare cells that are less than 0.1% of the population. And although our focus is going to be on the Paula today, if you want information how the Paula can be used in single-cell omics and bare cell isolation, we can also talk about that and share information. But before I go any further, I wanted to check that does anybody here have experience in generating cell lines? Yep, I see some hands coming up. Great. So how are you currently doing that? Are you using limiting dilution pipetting? Anybody? Are you using FACS to isolate single cell clones? I see some hands. Great. Does anybody have experience with the Paula? Not yet? Okay, well, I hope to convince you that you should give it a try. Or perhaps some of you have been spared the pains of single cell isolation so far. As we heard from Dennis's talk, generating cell lines is a lot of work. And no matter what method you're using to edit your cells, you know there's a lot of planning, and you heard that from Dennis as well. A lot of planning before you even start optimizing your transfection conditions and then finally transfecting. Then you still have to isolate those clones, analyze your clones, and before you can use them for experiments. And about 50% of surveyed CRISPR users identified this clonal isolation step as the most laborious of them all. And that is probably because the traditional methods of single cell isolation, which are limiting dilution and FACS, do have some drawbacks. First of all, limiting dilution pipetting requires you to dilute your cells to about half a cell per well, which means that at best you can expect to get about 30% of your plate to have single cells. FACS is obviously more accurate, but it can actually exert a lot of pressure on your cells, which can kill a lot of those cells, especially more fragile cells like iPSCs. And as a result, most researchers are used to plating tons of plates. And then the plating part may not be that difficult, but then you're actually think about the work that comes after screening those plates, imaging them. It's a lot of work. So that's where the Paula comes in. It is much more accurate than limiting dilution pipetting. You can expect to get more than 80% of your wells with single cells. And like FACS, if you're able to label those cells, like Dennis mentioned, you can also use that fluorescent signal to isolate only the cells of interest. Unlike FACS, however, it is more gentle, meaning that you're going to get a lot less cell death. And as a result, instead of generating and filling up a bunch of plates, you can maybe just fill up one plate. So less work, more clones. So I'm going to recap a little bit. So limiting dilution pipetting, it is slow and inaccurate, and it does not have that fluorescence detection. Bax, on the other hand, is pretty harsh on your cells, uses high pressure, and causes cell stress. And if you ask me, operator stress. It also requires some high-level training. So typically there's a dedicated person operating the Bax instrument, and there's a high risk of contamination between samples and just keeping those cells sterile. Paula, on the other hand, is gentle on the cells. It's very easy to use. It's fast, it's affordable, and very little maintenance. There's a lot of advantages for Paula over Bax, but I'm just going to point out a couple here that our customers really like. I've talked about that pressure, so we use less than 2 psi, whereas typically Bax instruments use at least 20 psi. You can start up a Paula in 5 minutes, and if anybody's planning on, hopefully some of you are planning on joining for those demos today, it can be started up in 5 minutes instead of that 45 minutes that it takes to get a Bax instrument up and running. People that are using the Paula for genomics applications really appreciate the 1 microliter droplet size. It is very consistent. And then people that are doing cell line development appreciate how easy it is to do sterile sorting. I have a quick video that hopefully works.
I'm Rachel Tallis, a field application scientist with Bio-Techne. I'm here to introduce you to the Paula, our automated platform for sorting and dispensing single cells into 96 or 384 well plates. The Paula uses microfluidic-based technology to gently and efficiently sort single cells. It's 20 times gentler than a traditional Bax sorter, ensuring better cell viability and clonal outgrowth, even for sensitive cell types like stem cells and primary cells. The intuitive design makes it easy to operate with no special training required. Plus, sterile, single-use cartridges sample contamination-free, even when switching users or samples. All this power comes in a box the size of a desktop printer, so you can easily move it into or out of a tissue culture hood. With 2 laser configurations, 488 and 405, or 488 and 561, Paula delivers up to 11 fluorescent channel measurements, making it perfect for applications like cell line development, single-cell genomics, CRISPR editing, antibody discovery, and verse cell isolation. The Paula makes single-cell sorting gentle, simple, and accessible. Discover how Paula can power your research at biotechne.com/Paula.
So to recap, the Paula uses 3 core technologies: microfluidics, flow cytometry, and liquid dispensing. You will pipette your sample into one of our cartridges. I will actually be, maybe I can I pass these around? Perfect. One, pass around from the other side as well. So you pipette your sample into a reservoir, and it's going to be guided through that microfluidic channel, and the cells are going to be interrogated by laser. And then light scatter and fluorescence emission signals are used to identify those cells of interest, which are then sorted into a plate using liquid dispensing. Here's a blow-up of one of those cartridges. So this is that reservoir where you pipette your sample, microfluidic channel in the goes down in the middle, and then your cells are going to come out through a nozzle at the end in that 1 microliter droplet. Your sample actually never comes into contact with the instrument itself, and that really helps ensure that that you don't have any sample-to-sample carryover. As Rachel mentioned in that video, we have 2 laser configurations for the Paula, the 405, 488, which is favored a lot by people that are doing single-cell genomics because it can do those DAPI stains, the common nuclear stains. Here at the core, you have the 488, 561 laser instrument, and we also have 1 demo unit here as well. The Paula has 3 sorting modes. The single cell dispensing mode is great for isolating either single cells, or if you need more than one cell per well, you can do that as well, into either 96 or 384 well plates. It does it pretty fast, in about 2 minutes for 96 well plates, and 384 well plates in about 6 minutes. The bulk sorting mode functions kind of like Bax instruments, where it's good for enriching a population of cells into a tube instead of into a plate. And we run it typically at about 20 to 50 events per second, but you can speed that up depending on how pure you want your population to be and or how much time you want to spend on sorting. The rare cell enrichment mode is very unique to us. It allows you to isolate very rare cells. So if you're ever dealing with anything like circulating tumor cells or fetal cells, this one allows you to isolate those rare cells. That one is only, you use a single laser mode for that one, for the enrichment step. The software interface is very intuitive, and hopefully you can appreciate that during our demo as well. The startup and shutdown procedures are automated, and you're going to be guided with these prompts that appear throughout the process. So let's go over some of the performance data that we have generated in-house. So here in this experiment, they dispensed HEC, CHO, and A549 cells and compared the accuracy of PALA versus limiting dilution. And this, I believe it was about 16 plates per cell type, so quite a few cells. And if you can see here, the PALA cells, these are dark blue, the PALA sorted cells, we saw about 80% dispensing accuracy of single cells. This was determined by imaging the plates after sorting, and very few wells that had either 0 or more than 1 cell per well. Whereas for limiting dilution, as you could kind of expect, you only get about 20 to 30% accuracy, and most of your wells are actually going to be empty wells. Another metric that we looked at is outgrowth. So how well do those cells grow out after you dispense them? So we let those cells from the previous slide grow for 10 days and then image them again and to determine which of those cells actually grew out. And limiting dilution is considered the most gentle method out there for cell line development. But with the PALA, we actually get very similar outgrowth as you would get with limiting dilution. So this really speaks to that gentle nature of the PALA. So I only showed you data for 3 different cell types, but our customers are using the PALA for lots of different cell lines, including very more fragile cells like iPSCs, obviously, stem cells. We have people sorting nuclei, so various types of cells. I now want to share some data from just a few publications from our customers who are using the PALA for cell line development. So this is a group from AstraZeneca. They are generating cell lines for therapeutic protein production, and they were using FACS originally, but they were concerned about that pressure that the FACS exerts on the cells and whether that has any issues with the cell outgrowth and how well those cells are functioning after sorting as well. So they also were looking for a more simple way to generate those cell lines. So they actually set out to compare the PALA with FACS, and some of the metrics they looked at were dispensing accuracy, how well, again, those clones grow out, and as well, the most important possibly being how well do those clones produce that protein of interest that they wanted to produce. So for outgrowth, they were actually dispensing into 384 well plates, and I think this 7,466 wells, I want to say that I did the math and it comes out to about 20 plates. They did see greater than 95% accuracy of single cell dispensing, and only about 3.7% of the wells had more than one well. For outgrowth, protein production, and doubling time, they also looked at 2 different CHO cell lines. These were CHO cells that were adapted to suspension growth, and they had an easy-to-express protein cell line and a difficult-to-express protein cell line. They did not actually see differences in outgrowth. They are very experienced FACS users. They also did use a larger nozzle size, 140 micron nozzle size, and not all FACS cores are able to do that. But when they looked at protein production with the cells that were sorted either by PALA or FACS, the PALA sorted cells produced a lot more protein of interest. And they thought that this could be because of the PALA sorted cells also were doubling slower. And this is in CHO cells is associated with increased protein production. The difficult to express protein also showed a similar trend, although this did not reach statistical difference, and there was a lot more variability in both the BAX and polysorted cells. But on average, the polysorted cells still showed more protein production. So in conclusion, they also looked at things like cost and things like that, and they concluded that the poly will allow for the selection of better producing cells. And so they are some very happy polyp users. This next example comes from our customers that are at Gladstone Institutes. They developed a workflow to produce enhanced iPSC-derived NK cells for cancer immunotherapy. The NK cells are known to target several cancers, but they don't tend to stick around for very long. So they generated dual edited iPSCs where they did a TGF-beta R2 knockout to reduce the TGF-beta mediated inhibition of NK cells in tumor microenvironments. And then there was also evidence for IL-15 that could improve the persistence and cytotoxic effects of these cells. So they also did a IL-15 knock-in. They then characterized the cell line and evaluated its cytotoxic activity. This is kind of their workflow. So they actually used PALA twice. They first knocked in IL-15. Then they used the PALA to isolate and characterize those iPSCs that had the knock-in. And then they went and knocked out TGF-beta R2 gene and verified that knockout as well. I'm only going to show very little of the data, but what they found is that in the cells where they knocked out TGF-beta R2 and knocked in IL-15, these cells persisted longer than the wild type cells. And when they did a cytotoxic activity assay, over here in red and black, you can see either in black, the wild type NK cells and the IL-15 NK cells had similar cytotoxic activity. And I'm not showing you the data here, but they also compared the wild type and the knockouts for in the presence of TGF-beta, which again inhibits those NK cells. And in those conditions, these edited cells actually did better. That was just a couple of publications. Of course, there are much more than you can look for. We'll be happy to share these as well. And now I want to switch gears a little bit and just give a very brief overview of some best practices, how you can also ensure the outgrowth of your cells. And I think we're all know that even hardy cells like 293s can be affected by stress. And these stress factors could include either overconfluency or undercrowding, any kind of media changes or temperature. Lots of things can affect even these cells. And of course, this undercrowding is specifically relevant to single cell cloning because cells communicate with each other and they don't like being alone. So they communicate with each other through a variety of mechanisms, including chemical signals and physical contacts. This allows them to coordinate their activities. And so to ensure optimal colony outgrowth, it's really important to reduce stressors, any kind of external stressors that you have control over as much as possible. And this includes cell health before, during, and after single cell sorting. Some of the ways you can minimize cell stress at each stage when you're starting with healthy cells. So that is a very crucial factor. So if you need to saw cells, you want to do that at least a week before you try to sort them so that they have some time to settle and recover. Also, if you're doing any kind of transfection, you want the cells to recover. We recommend at least 48 hours, typically a little bit longer after transfection or electroporation. And then if you have adherent cells or suspension cells, you want to make sure that they're in log phase of growth. And for adherent cells, that's typically 70 to 80% confluency. When you're preparing to sort, it's also a good idea to minimize the time that those cells are out of an incubator. So you want to prepare all your reagents beforehand, make sure everything's ready before you dissociate your cells. when you resuspend your cells with the pola, you can actually resuspend them in media with protein in it. So up to 10% FBS is totally fine with the pola. That's where the cells are happiest. So that's what we always recommend. Also, we recommend filling out your plate for media already beforehand as well and put that in the incubator so that can stay warm when you prepare other things. A key factor is also that we recommend is using a live dead indicator because you want to be sorting only those cells that are alive. And if you ever need recommendations for viability dyes, feel free to reach out. Also, if you are doing any kind of staining for your target cells, we recommend that you optimize those staining conditions and identify any controls before you start your actual sorting. On the day of your sorting, you want to make sure that you prepare a good single cell suspension. A lot of people that work with iPSCs are probably more familiar with clump passaging, but for single cell sorting, we want to ensure that those cells are well dispersed. So we do recommend using a filter to remove any clumps. Again, you want to resuspend your cells in media and you want to count them. And for the pola, a sweet spot for dispensing them as single cells is about 10,000 cells per mil. That helps you achieve the optimal rate for dispensing. Of course, if you were to use the pola, we would go over these in more detail, and we'd be happy to help with any kind of training if you needed that. After you've sorted your cells, you want to put that plate back into the incubator as soon as possible and just leave it there for at least 24 hours untouched. Also, if you want to image that plate, we recommend that their cells can be really hard to find in that three-dimensional space, that we recommend either letting them settle for a little bit or giving the plate a quick spin before you image them. As a reminder, some cells just really don't like being alone, so conditioned media at 1-to-1 ratio can sometimes help some cells, and sometimes increased FBS can also help. I've heard from a customer who's working with HeLa cells, and you would think that they don't require anything, but they actually did see better results after they used conditioned media. Of course, that could be just dependent on what kind of a gene you're editing as well. We do have a wonderful workshop from my colleague Celeste on our Bio-Techne Academy that is free. So she will, in her workshop, she will go into more details about how you can gate the best practices in gating and making sure that you get those single cells. So Bio-Techne Academy, you can sign up for free. It has a lot of other resources for our other instruments as well. So in review, Pala allows you to isolate single cells in minutes instead of hours. It's much more gentle and you typically get better outgrowth. It does have more fluorescence options than none with limiting dilution. So you have that ability to select your cells of interest, and we can do lots of different cell types. With that, I think I'm going to pass it on to Ryan